going to do the properties of acids lab. We want to see what's the same about different acids, see if there's any common properties. So the three acids we're going to use today are hydrochloric acid, sulfuric acid, and acetic acid. Okay. We're going to test them with 7 different substances to see how these actually react. So we're going to use some magnesium, so you can see that silvery metal in there, an indicator called bromothymol blue, universal indicator, phenolphthalein, red litmus paper, and blue litmus paper. And we're going to use sodium bicarbonate, or the more common name is baking soda. Okay, so I'm going to put these in order so they're in the same order as on your lab. And what we're going to do is we're going to test these. So since we're testing for 7 different substances, I'm going to put sulfuric acid in 7 spots across the spot plate. And then I will put hydrochloric acid in the next 7, so row 2. And then we'll put acetic acid in our 3rd row. Okay. Okay, so sulfuric, hydrochloric, and acetic acid. So what we're going to do is first we're going to test it by putting some magnesium ribbon into our acids. I'll drop it in there, and we'll probably have to zoom in pretty close to see if there's a reaction happening. Okay, I'm going to make sure they actually get under the surface so that we can have that reaction happen. Sorry, I gotta use a different one just to make sure we're not contaminating the samples. Okay, so when we take a closer look, hopefully you can see the sulfuric acid with the metal. You can describe what's happening there. We have hydrochloric acid in the metal, definitely a reaction there as well. And we have acetic acid in the metal. So that looks like they're all reacting the same way. So you're going to want to put your observation down in the chart. So the next thing that we're going to check is we want to see what the reaction is like with bromothymol blue. Okay. So you can see it's a blue color before I put it in. Okay, and we have an instant color change. We can maybe see the color change a little bit better if I put a piece of paper underneath. Okay, very, very vibrant, like a dark yellow, almost an orange color. Okay, very yellow. It's very concentrated, I think. Okay, so next we're going to try red litmus paper. And we call it red litmus paper, but it actually looks kind of pink. And so we'll put it into the spots and see if anything happens. Okay, so they all look the same. And just for reference, there's our original piece. I put one outside of the plate. So we're not really seeing any difference there. So for our next test, we're going to use the blue litmus paper instead. And so this blue litmus paper, you can see it on the counter. I'll put one in front again so you can use that one for reference. So putting the blue paper in, and we are getting a reaction this time. Okay, so that does not look the same. Okay, so whenever we use litmus paper, we either describe it as red or blue. So if it looks pink, it means it's red. Even if the blue looks a little bit, it can have a bit of a purple tint to it, it's still blue. Okay, so either red or blue. Those are our two colors that we use for our observations. Okay, universal indicator. So, see what our results are like for this one. So universal indicator is kind of a green color in the bottle. Okay. And when it goes into our spot plate, we have a color change again. And just to make sure you can see it well, put the paper underneath it. So we're actually getting a red color. Okay, so we still have phenolphthalein to check. It looks like a really big word on your page, phenolphthalein. So, a little bit in each of these. And so you can see that in the bottle, it's clear and colorless, and there's no real change, okay? These are the ones that have the phenolphthalein in them. So now we're going to try a little bit of sodium bicarbonate or baking soda. Okay. So I will try to put a little bit into each one. So hopefully you can see that. So that's with the sulfuric acid. Lots of bubbles. And now it's all reacted, so you can still see the bubbles that are left over. Oops, I definitely put more into the second one. So it's bubbling. We may not see it all disappear just because I put way too much in that one. And our bottom one, the acetic acid, right, that one also had the proper amount in, and we can see it bubble really quickly and then go away.